And the mic on? All right, it's on. Welcome back, everyone. We have reached to the final session of the day. In this session, our esteemed panelists will discuss in depth advances and challenges in vaccine development and manufacture. To start, it's my pleasure to welcome the chairman of the session, Dr. Jamila Al-Wahed, the Vice President and Head Global R&D, JSK. The mic to you, Dr. Jamila. It's a pleasure to have you with us in Riyadh Global Medical Biotechnology Summit 2021.
Good morning for those that are based in North America. And salam alaikum for this meeting. I really feel honored to not only participate to this prestigious summit, but also to be able to chair this panel with really a knowledgeable speaker. So vaccines has never been as important as the last 2 years. A world without vaccine is not a world. And this is something that the pandemic has taught us. Across the speaker and the discussion we will have during the panel, we will understand what are the challenges ahead of us, but also making also some highlight about what were the successes of the vaccinations throughout the last century. I will be joined by people coming from different sectors. So Jan Enrich coming from Sanofi will give us some insight about the influenza vaccines. Dr. Hani Al-Shani from Pfizer, who will be discussing about biotechnology in vaccine development. Naif Harbi will discuss about the vaccine R&D in KSR, the experiences and the challenges. And Sven Free will give us insight about the corporation and vaccines in Danaher. So without further ado, I want to welcome the first speaker, which is Hani El-Hashmi, who will give us some insight about biotechnology in vaccine development. Hani, the floor is yours.
بسم الله الرحمن الرحيم والصلاة والسلام على سيد المرسلين سيدنا محمد وعلى آله وصحبه أجمعين. Thank you, Mr. Chairman and panel for the introduction. First of all, I would like to thank Kmark and National Guard for a very elegant conference so far. Grabbed attention from all over the world, and I need to speak about that. I was just talking to Dr. Ahmed. There is about 10 to 16,000 attendees per session for the past day and a half, which talks about the interest that actually this global summit has actually put into the community, and we would like to see more of this. So the task that I was asked to talk about is actually biotechnology in vaccine development, and I would like to take you through a journey just to make sure that the setup and all the understanding that is going to come after this presentation makes sense to you, and to connect this with our real life and what we have seen over the past as far as development and where are we expected to head to and lead to in the future. So this is my disclosure. So I'm Senior Medical Director for Pfizer. Presentation, this is just a disclaimer, so it's actually for education and only, and it represents my personal view about biotechnology and vaccine development, and I hope that you'll enjoy it. So just to be very simple, simplicity is the ultimate sophistication, as they say. I would like to make it really simple in the way that we actually go through this presentation. So I'll talk a little bit about biotechnology. I'll talk about vaccinology. I'll talk about both together, the combo power, as we say. And I'll end up with a small essential steps. So this is the essence of my presentation, where I think we probably, if I'm going to give advice for where are we heading and where should we go to, that will be my final slide. So biotechnology, if we talk about biotechnology as a term by itself, it's been first described by Carol Erke back in 1919. So that was more than 100 years ago. And it was briefly to describe new product from raw materials. And that happens with the aid of an organism. So a process or a system or an organism that can actually give you this new product. So if you think about what biotechnology in a very simple layperson understanding, so you have a new technology application, and you get a biological system or a process or an organism, you apply this biotechnology to it, and you end up with a new product or an improved product that of essential interest to many people, like pharmaceutical, livestock, and probably sometime even for the society. And when the society talk about biotechnology, they talk about biological manipulation to serve societal needs. So this is what the society need, and through my presentation, you'll understand why I'm saying society. So this is where biotechnology started. So we're talking ancient, and actually talking about pharaohs and ancient Chinese, and years ago, they needed biotechnology to actually start living their life. So they started with livestock, and they were able to move from domestication to fermentation. So they needed animals to actually start their life. So they started the domestication, and then after that, they started some form of fermentation, which is actually using the technology to help with their products. So they were able to bring cheese and different kind of drinks and so on. So this is very nice example of where things are actually heading. So if you think about biotechnology started from domestication, fermentation, we leaped into, in the 19th, 20th century to DNA, and people started to know more about the DNA level. Monoclonal antibody, PCR, transgenomic, and actually cloning. And recently, over the past 20 years, we were talking about the human genome. And what's next to come is very difficult to predict, because the technology that's being developed as we speak is actually going to serve us to know more and to do more. However, if we talk about medical biotechnology, I think that was the start. So it was basically by Herb Boyer and Stanley Cohen back in 1973. Those two actually, for those who don't know them, they were actually the founder of Genentech. Genentech is actually a small biotech company, was acquired back in the 1990s by, actually partially by Roche, and then fully is actually being acquired now by Roche. But their start of the biotechnology was actually dated in that day, 1973. What they did is actually they were able to introduce DNA into the E. coli and actually create a new transgenic bacterium. With that, they were able to actually start producing new product, new protein, such as the insulin, and the technology just expanded after that manipulation. So DNA biotechnologies was actually the start, and was back in the 1973. Based on the work that was actually done by them, 3 different physicians, one from Switzerland, 2 from the state, from Johns Hopkins, were able to get a Nobel Prize a few years following that, just because they were able to discover the enzyme, the restriction enzymes, that was responsible for the molecular genetic changes that they were able to do. So the application of that restriction enzyme was actually the key factor for unlocking the future for everything that happened afterward. So moving to vaccinology, and if I talk about vaccine these days, everybody probably know a lot about what vaccine is, but basically administration of antigenic materials. And this antigenic material is considered to be the most effective way of disease prevention and actually control of any pandemics. Continuously evolving science, as we also have seen over the past year, the development of different vaccine and how is the speed that happened in the past year, and the technology that evolved, and actually the collaboration that happened that actually catalyzed this speed. So the vaccine basically is a biological preparation that is provided to acquire an immunity. So acquired immunity that we're talking about, and the material that is being actually given is resembling the disease-causing pathogen or the disease-causing antigen. And when I say antigen, meaning that because we're not talking now about infection and infection control, because the wide spectrum for protein production, we're talking about all kind of disease prevention. So we have known actually in the past inactivated microbes, attenuated microbes, toxin, and so on. So history of vaccine also is very important to understand and to know. It began with actually infection disease being spread in the human early years. So we knew about it because of the example that had been described in different society in the past. And the evidence from the Chinese is actually very interesting, because they probably were the very first people who actually gave vaccination. And the method they use is actually virilization, basically blowing the particle into the nose of the kids. This special virilization technique was actually also described in Turkey and also in Africa before it spread to Europe and eventually to America. But the real story of the vaccine started with Edward Jenner, and this name is very important because he is considered to be the father of immunology, if you were to say. What did he do? Everybody knows smallpox, judari. This is actually a disease that killed more than 300 million people. 300 million people in the 20th century. So no wars have ever done that. So 300 million people died because of this disease. But his actually work dated back to the 1700s. So 1796, he was able to innovate a small, he did a small kind of test and experiment, and he was able to actually use this into one of the kids in his garden. So they were able to create this technology and brought something, we call it actually the concept, the proof of concept. So I don't know if people have seen this picture before, and this is actually one of the very disturbing pictures about vaccine resistance or anti-vaxxer. So his method was actually developed and was actually even improved, and the smallpox was completely eradicated in 1979. But we have still that actually period of time between the 1900 and until 1979, there was a lot of issues with the anti-vaxxer, and one of the pictures that is actually very famous for people, and it's already been published in different articles, is actually the two kids. One received the vaccination and the other one did not. And unfortunately, a bad outcome has happened to people who did not get the vaccine back then. So the story continues. So rabies disease was actually one of the things that people wanted a vaccine for. And we see here Louis Pasteur in 1885. He was able to actually generate such a very important vaccine, and that impacted the human diseases and human nature and actually the science at the time. And following that, a lot of other things had happened. So we have seen antitoxin vaccine against diphtheria, tetanus, anthrax, cholera, plague, and so on. So there is a lot of things that has happened after that, and we're seeing more. So what happened in the 20th century? Probably the middle of the 20th century was the very active time for vaccine and vaccine development. Methods of growing viruses inside the lab have actually become more and more standardized, and people are able to do it. And that gave a space for scientists to start creating more and more vaccine. One of them is actually a very important vaccine that we know of is polio as well. However, when we talk about biotechnology and the modern biotechnology tool and being able to unlock the genome and knowing exactly what the genes that actually being harbored inside it, brought a different angle to vaccine and vaccine development. So the new biotechnology that may result in production of therapeutic vaccine which are effective for all diseases. And when I say effective for all diseases, it actually expands beyond, as we say, infectious disease. So when we employ the biotechnological innovation, this is going to unlock a lot of futures for scientists, and that's actually currently happening, and we're probably going to see more of in the future. So when you bring the two together, the medical biotechnology and the virology as well, there is three basic concepts. So the first one is actually the separation of a pure antigen, and that pure antigen can be used, can be actually purified using monoclonal antibodies. So the actually purification, separation of specific antigen. Sometimes the second actually important aspect is actually the synthesis of antigen with the assistance of a clone gene. And the third method is actually synthesis of a peptide of a total protein that can be used as a vaccine. So if you want to summarize biotechnology in the vaccine development, maybe those three very basic concepts need to be kept in place and kept in mind. And this is kind of a holistic picture of vaccine and biotechnology have been used in vaccine development. You can see the toxoid, the polysaccharide, and the whole pathogen, and so on. And you can see in the very bottom there the potential future development using actually some form of nucleic acid vaccines or vectors vaccine or conjugation. But what I'm going actually to spend some time on over the next maybe 10 minutes or so is to give you some example of important biotechnological methods that's currently available and probably available, I would say widely available, because some of them are actually basic science kind of 101. So the first one that I probably will shed some light on is something called reverse vaccinology, a simple concept applied in vaccine development, and it gave us what we know now as COVID vaccine for what it's worth, and many other examples of vaccine. Recombinant subunit vaccination, recombinant protein vaccination, and basically nucleic acid vaccines, such as the DNA vaccine and the ribosomal messenger RNAs vaccination. So reverse vaccinology, the very basic idea is actually with the word itself, reverse. So you go with a full genome sequence of a specific pathogen or a specific virus, and this actually genome sequence, you can analyze it with some form of informatic and bioinformatic method, and you'll be able to explore all the genes that's encompassed within this gene sequence. And being able to do so, you'll be able to detect all kind of antigen, potential antigen that can be used as a vaccine in the future. In this diagram, you go from the very basic full genome sequencing. You know everything about the genome. You'll be able to actually create some idea about potential vaccine and protein that you're going to use, and you shortlist the vaccines, the potential to number of candidates, and you start your actually pre-clinical and clinical trial as far as the investigation goes. So it did change the concept of vaccine and vaccine development, and we've seen that, we lived that over the past many years, and probably more so over the past 2 years. The list of the candidate, the novel candidate that can be given after the assessment of the genome becomes very manageable. So rather going through thousands and thousands of potential vaccine and target, you actually shortlist this. And by applying this approach, you'll be able to actually reduce the cost of making the vaccine and the vaccine trial, and you'll be able to actually shorten the time from the preclinical to clinical trials. So the recombinant vaccine subunit, or recombinant subunit vaccination, so basically the gene cloning is the powerful tool that we're trying to use, and the recombinant subunit typically use actually a fragment of the protein, which is pretty much the antigen, and express this in the laboratory using viral DNA. The gene that codes for the antigen is inserted into actually a host, or in most of the cases, a yeast genome, and the final product gets purified, and you end up with a pure protein that's ready to be used as vaccine and can be taken into the next level. This is, again, an oversimplistic view of this approach. One of those approaches is actually hepatitis B virus. You take the antigen, you clone, you actually get the sequence specific, the gene that you're interested in. You put that into a host. You put that into a yeast. You grow it in a fermentation tank, and then after that, you purify the product, and you end up with a small product that can be used as vaccine to start the clinical trials of so. The protein vaccination is very similar in terms of the idea. But I'll move here to the DNA and the RNA vaccination. And if you can see with this diagram, it's again an oversimplification of the idea. So you have either the DNA or the RNA. This gets injected. It's actually being transformed into, it actually can result in an immune response. This immune response can actually give us what we look for as either humoral immunity or actually antibody-mediated immunity as a basis of the DNA and RNA vaccination. So what I would like to focus over the final few slides, if we talk about the biotechnology and the vaccine biotechnology in terms of DNA and RNA, people think this is actually something that happened over the past year. This has been published and studied for the past at least 15 years. What I'm showing you here is actually it's actually a review article published in 2012 and speaks about everything that we've been asked to believe today about the safety of the vaccines. So those vaccines are actually fairly safe. People knew about them. In 2012, they actually published a review article on those things, and we're trying to convince ourselves about the safety. So moving with that, this diagram I took from a textbook published in 2017. And you can see from this textbook, DNA vaccination, messenger RNA vaccination, no commercial product was available for human in 2017. So today, I'm sure everybody knows that this is not the case. So life is changing, and science is moving forward as well. So a very good example of the collaboration that happened during the pandemic period. Early in 2012, in December, January of 2020, late 2019, the sequence of the COVID was actually published online in the library online, so people have the availability, and all the scientists were able to take the sequence and start working collaboratively on different platforms of the vaccination, whether it's mRNA, DNA, viral vector, and protein. And that's why we moved very fast from the preclinical to the clinical, and built up world. Life will go back to normal, and this will be history behind us, but we learned the lesson. So what I'm trying to say with this slide, this is my last slide, and I think it's very essential steps. And we always focus, when we talk about biotechnology, on the non-human infrastructure requirement, where actually the opposite is actually very important. So you need the right advisors, you need the right team in your team, you need the right collaborators. And those three are actually human factors. With the most important criteria for those human factors is actually credential and track record. Because if you have an advisor that doesn't have the track record or the credential, not sure if you're getting the right advice. If your team members don't have those two, then maybe they're not the best team member. If the collaborator that you're going to collaborate with don't have the credential, don't have the track record, then probably this collaboration is not the best collaborator. Definitely you want to look for the right project for the right time, and you really want to have your right timelines. Instead of saying, I want to do this over a period of few weeks, maybe the right time is actually a couple of months or maybe a year. So those 5 rights is very essential to develop a biotechnology. And if we're talking about the future, I think the future lies within biotechnology. I'm glad to see such a big event happening here in Riyadh as a global summit, because this understanding and awareness about the biotechnology in the region need to be enhanced, and people need to start collaborating with each other to be able to leap into the future and join the industrial life. And thank you very much with all different language, and in the middle, shukran jazeelan.
Thanks a lot, Dr. Hani El-Hashmi. That was really a nice overview about the story of the vaccination and also the big inventions that has led to the different technology that is being used in vaccines. I will be giving some insight to complement what Dr. El-Hashmi has presented about what GSK is doing in the field of vaccines, and what will be the future about vaccines in line of what are the medical needs, but also the technology that now is being available to us. So a few words to remind the value of vaccines, which we tend to somehow forget because of the success of vaccines. We have now in front of us hesitancy of vaccinations, and the reason is because people have forgotten some deadly disease. According to the WHO, the World Health Organization's only clean drinking water rival vaccination in its ability to save life. Vaccination across the world saves 2 to 3 million deaths every year, save around 800,000 disability in children, and provide also the opportunity to have a longer extension during the life. So those are facts that are database thanks to the vaccinations that people should remind whenever we have in front of us people that are hesitating against the value of vaccines. So just a couple of examples about what has been able to achieve through vaccination across the world. Diseases like polio is almost eradicated with almost no cases. Only few cases appear in some countries, but overall the vaccination has really done a very great job. Rubella, pertussis, diphtheria, tetanus, you see that all those vaccinations that has up to 99% of efficacy was able to dramatically impact the disease caused by those viruses. So now, if we zoom and look at what are the specificity of vaccines and at GSK, the biggest organization of GSK vaccine is based in Belgium, with sites across the globe. But the biggest footprint is in Belgium, where we have all the end-to-end activity linked to vaccine development, meaning the R&D, the manufacturing and packaging, and the distribution across the world. It's important to understand that one of the big challenges of the vaccine is the equity around the world, and we have seen through the pandemic how much is important. Equity means also making sure that the value and the price of your vaccine is also adapted for developing countries or industrialized countries. The model that GSK has really worked on is to have tier pricing, where around 70% of the vaccine that we produce go to the developing countries to make sure that we have equity in our development. So what it means also in terms of infrastructure to develop vaccines. We have heard about the different technology, but the manufacturing infrastructure that is required for vaccine is quite important. And you have here the largest production site that is based in Belgium, which is the biggest in the world, where most of our vaccines are manufactured. So we produce around 2 million vaccines a day that are distributed across the world. And why this infrastructure is important, it's because all the quality tests that needs to be done, the scale up that is required to face the supply needs are quite important. And that's why in the past, there were only few players in the field of vaccines because of the huge amount of investment that is required for manufacturing of vaccines. Quality of the vaccine is also something which is specific because we inject vaccine in healthy subjects and in babies, children. So you cannot afford any side effect of your vaccination. This is why quality and the safety is quite important. And it's important also to remember everyone that the medium framework for developing vaccines was around 20 to 25 years in the past. We have heard in the previous talk that now we can accelerate, but that would not have been possible when we speak about vaccines that go into babies, because you have all this safety that needs to be assessed properly to avoid side effects of those vaccines. So we have also the R&D. We have essentially 3 sites. The biggest one is again in Belgium, so you have it in the middle. And you have a site in the US, Rockville, as well as in Siena, where we do all our discovery and research. Something that I have heard a lot during the summit, which is quite important, is the public-private partnership. And I want to highlight that GSK, for most of our pipeline vaccines, has been done through collaborations, which again highlight the importance of this collaboration between research and also private companies. So now, if we go back to the history, and I will not repeat what has been just said before, the only part that I want to insist is that technology has been quite important in the history of the vaccine development, starting with very empiric vaccine like Jenner, where you have inactivated viruses injected through something which is now genetically coded vaccines. And all those new technology, as they were developed, they have been able to unlock the possibility to develop a vaccine to tackle a specific virus or a bacteria. Now, again, so just here to remind everyone about the technology and the approach that has been able to unlock the vaccines. The other element that is quite important to remind everyone is also when we do a vaccination, to basically educate your body to be able to mount an immune response, to be able to fight if you encounter the virus or the bacteria. What we have learned throughout the year, through the science, is that our immune system is not the same when we're born and when we go elderly, as we evolved. So this is important to remind everyone that when we develop vaccines, it's important also to realize what are the target populations that we are facing, because a vaccine that you develop for a baby will not be necessarily functioning for an elderly because the immune system is not the same. And what we have learned is that the period where the susceptibility is quite high is where you are born, where your immune system is immature, and usually vaccination do a great job there. But as you get older, you are also aging your immune system, which is called immunosenescence, and you need to have a reboosting approach of your immune system to be able to be less susceptible to infectious disease. And that's why we hear more and more now that there is a need of have a life course immunizations to be able to protect not only our babies, children, but also to be able to protect our elderly. And again, if we do the parallel with what we have heard about the COVID-19, the more susceptible population was our elderly population. So can I have the next slide? So it seems that I have some trouble. Can I have the next slide? Okay, so now what is the vaccine innovation trend? I think we have done a great job in prophylactic vaccines. There is now more than 22 vaccines available against preventable diseases, but there are still some medical needs that needs to be tackled. We also have much more tools and understanding in terms of technology to understand the viruses, the bacteria, and the parasite, to know which antigen we need to tackle. And also, we had the opportunity to leverage the live technology and data that is now available to be able to discover new vaccines against preventable disease, prophylactic. The idea also is to use vaccine approach to tackle therapeutic vaccines, which means that not only you are using this methodology, which is the vaccinology, to prevent, but you could also think about treat people that are infected by a virus. And as an example, the hepatitis, which unfortunately, even though there is a preventable vaccine available, the coverage across the world is not optimal, which means that there are people that get infected by hepatitis and have a big chance to develop a cirrhosis, and that could eventually move into a cancer. So being able to use this approach as an immunotherapy vaccine approach will be important. And lately, what is important is what we call a life course immunization, is how can we also make sure that we provide the right solution to our older adult populations. They need also to have a protection because they are more susceptible to infectious disease. So if I can have the next slide. So you have here a summary of what are the remaining medical needs, which is, I would say, quite high, without naming tuberculosis, the respiratory syncytial virus, the malaria. Even though we have now a vaccine which is has been launched, we need to continue to fight. And then you have a lot of new bacteria where we are facing potentially the next global threat because we don't have antibiotics that are now being available to tackle some resistance in this, for this bacteria. And the only weapon that also can be investigated is being able to have vaccine developed against those bacteria. Next slide, please. So what are the, can I have the next slide? So among the tools, we have been talking about whole cells bacteria, inactivated viruses, recombinant proteins, but there is also other element that also is quite important in the field of vaccinology, which are called adjuvant, which has been shown to be very efficacious to tackle some disease. For example, malaria, the first vaccine that is being developed is using this technology to be able to have the right protections. It has been very useful in the elderly population where you have this trigger of increasing the immune response induced by the vaccine is important. So that is something that is a technology that is also of great value as we move forward for new vaccines. Next slide. One example, if you think about the last pandemic in the flu, H1N1, we have used an adjuvanted vaccines. And the beauty of having these adjuvanted vaccines was not only on one hand, you could decrease the amount of antigens, being able to supply more the world on vaccines, but also you induce a more diverse immune response that is able also to protect against several serotypes from the flu, which is seen here, where we compared a plain flu vaccines versus an adjuvanted vaccines, where we have seen not only a good immune response across age, but also across protections. And cross-protection is also very important. If you think again the parallel with the COVID-19, being able to have a cross-protection against a different variant is something that you aim when you are developing a vaccines. Next slide. So what are the remaining, and I will give a few examples of projects that you are working on where there is an important medical need. If you think about the respiratory disease, after the flu, one of the viruses that is causing high hospitalization and low respiratory disease is the RSV virus, so respiratory syncytial virus. The two populations that are sensitive to this virus are the very young babies and the older adult. When we get more at the adult age, usually our immune system can be able to function and fight against the virus. But those young and elderly, they need to have a solution to be less sensitive to these viruses. The way we approach it, next slide, is to design specific vaccines that allow to cover those two populations. One is what we call a maternal immunizations, where you vaccinate mother at the third trimester, inducing antibody response that will be transferred through the cord blood to the baby. And as the baby born, he will have antibodies to be protected against the viruses. This has been shown already for the flu. And we have also a nice result that show that if you have the right level of antibodies in the babies, you are protected. For the elderly, you have to tackle an aging immune system, and your vaccine needs also to be addressed to cope with this immunosenescence. So for the elderly, we are developing an adjuvanted vaccines to be able to protect against the respiratory virus. Next slide. So the last class of two vaccines that I would like to highlight, again, one of the success of the vaccination across the last century, the vaccination was the only approach that has allowed to prevent resistance to antibiotic. If you think about pneumococcus, if you think about pertussis, data has shown that this is one of the tools, one of the weapons that has avoided to have resistance to antibiotic. For bacteria where no vaccine is available, you have now, we have to face those resistance to antibiotic with less and less small molecules or antibiotic available to treat infected patients. That's why, as part of the effort across the world that is going down to find new antibiotic vaccines, antibiotic, but also monoclonal antibody. Vaccine has been put also now into the table to be able to find eventually a solution. Next slide. The two bacteria where a lot of effort now is being done in GSK, but in other companies also, is the Staphylococcus aureus and Clostridium difficile, which again are bacteria where usually you see recurrences in those patients that are susceptible. And we see that there is also more and more resistance coming across the world against those two bacteria. So having in the future a vaccine for those patients might help us to give them some new opportunity. So if I can have the next slide. I'm sorry, but I cannot switch. So this is the example. We can go fast because we are running out of time. The last part of innovation that I wanted to highlight is the therapeutic vaccine, where you are not anymore in an intent to prevent, but rather in an intent to treat patients. And again, immunotherapy has been booming the last decade in oncology, but also you could think about new treatment in inflammatory disease. And the idea would be to look at how can we tackle through vaccination diseases, chronic diseases that are caused by viruses or other pathogens. The one that now is being developed at GSK is a therapeutic vaccines against tuberculosis in collaboration with the Gates Foundations, and also the therapeutic HBV vaccines to have a solution to treat those patients. So I would like to conclude, if I can have the next slide. Can I have the next slide, please? So if you think about what the vaccination has done the last, I would say, century, we have done a fantastic job in preventing disease in infants, where we have a weapon, I would say, that has allowed to really decrease substantially the number of deaths. Next slide. We are now working more and more across the industry to develop vaccines that are tailored for elderly patients. And we have seen a lot of recent advancement, like vaccine against the shigella disease, COVID-19, the flu vaccines. But next innovations where it needs to come, it's to be able to have an engine, a strategy to tackle diseases that are endemic in developing countries, to tackle viruses that are associated with chronic diseases. And one could dream also that eventually in the future, we could use the approach of immunizations to tackle chronic diseases and eventually cancer. And if you think about the next or the current big innovation in the vaccine is the mRNA platform, and Pfizer and Moderna has been pioneering into that. Those technologies were coming from the cancer vaccines field and were able to be scaled up for the COVID-19 pandemic. So this is the power of being able to understand the immune system and how you want to manipulate to be able to have new treatment in the future through immunotherapy. So from empiric approach, whole virus or inactivated, to a digital vaccine, which is what mRNA has been able to achieve lately. It's a wonderful journey about innovation, technology. And I'm sure that with the new life science and the understanding of the many pathogens that could evolve in the future as a pandemic, we do have, if we collaborate properly, the weapon to be able to fight any future epidemic or pandemic that might arise. So I will stop here. And now I will give the floor to the next speaker. So the next speaker will be Naif El Harbi, who is heading the vaccine R&D in KSA, who will speak about experience and challenges in vaccines. So I'm really looking to hear your talk. So the floor is yours.
All right, thank you so much, Jamila, for this introduction. I'm very glad to be here, and we're in KMARC. We're very glad to organize this summit to bring all stakeholders globally and nationally. I'm just waiting for my slide to appear before I start. And let me introduce myself. I'm Naf Al-Harbi. I have a PhD in vaccinology and molecular virology. And I've spent 6 years in Oxford University in the Jenner Institute in the vaccinology area, where we do from bench to animal to phase 1 clinical trials. And in the past 5 years, I've been in KMARC proudly trying to push this boundary in vaccine development. And if there is one thing that I can summarize with vaccine development, I would say it's very much easier said than done. Anyone could say, okay, you pick up an antigen, you put it in whatever system, you produce it, and here you go, you have a vaccine. But it's not as easy as that. And vaccinology particularly is multidisciplinary subject, but also it's multidimensional. So you will have business side in there, you will have politics in there, you will have regulatory, you will have ethical consideration. So all of these facets of complexity, which makes it very easy said than done. Can I have my slides? Give it to Khalid. Okay, so sorry for this delay. So this presentation meant to basically summarize what have been done in vaccine development in Saudi Arabia in the past 30 years, I would say. I will not focus much on the industry side of vaccine development in Saudi Arabia, because yesterday we have heard in the first session that some information from the Ministry of Industry about the capability of the country, different pharmaceutical companies existing and their capacity as well. So academically, this slide shows 4 papers. The oldest paper I could find in some academic institute in Saudi Arabia producing a vaccine was in 1989. And that was from Jazan Hospital trying to optimize the vaccine of pertussis by some method of inactivation. Another paper from Prof. Fahd al-Majhadi that was also on influenza vaccine based on DNA platform. And another paper in the 90s, early 90s, developing a camelpox vaccine. So this is what you can find in the 90s and 80s. Until recently, our vaccine academic effort was driven only for veterinary vaccines, especially after the Rift Valley fever or the MERS-CoV. So what we can say is our academic institution, when it comes to vaccine development, they're proactively responding to the national public health needs, mainly Rift Valley and MERS-CoV. And in this slide, we have, in the last maybe 4 or 3 years, we've seen more research coming into publication from various universities in Saudi Arabia, King Abdulaziz Imam Abdurrahman and K-Mark as well. Again, most of these papers, although a high-impact publication, they're focusing on MERS-CoV vaccine development. We've seen viral vector-developed vaccines for MERS. We also contributed to some of this work in the camel as intermediate host between what is supposed to be bat and human for MERS-CoV. So this summarized the vaccine biotechnology, although Dr. Hani Al-Hashimi explained it very well. What I just want to highlight here is that people moved now from conventional vaccine development into novel, where you don't use the whole organism, you use part of it, as he explained. But I would just want to highlight one thing here, that the conventional or the traditional notion about vaccine development is if you have the whole pathogen, you will have more higher efficacy, but lower safety. And if you go below until you get like only the DNA or the RNA, you will have higher safety, but lower efficacy. But this was not the case with COVID vaccination. We have seen that the new technology complementing, supporting the mRNA vaccines like nanoparticle encapsulation enhanced the efficacy of this technology while accounting on their safety profile. So this is the typical vaccine R&D pipeline. You'll have the discovery stage feeding into preclinical setting, and then phase 1 and 2 and 3 before regulatory approval and marketing. And this is all usually pushed by technology and by management and fund, but also it is supported by the public health need and the market. And that's what we've seen in MERS-CoV. There is a lack of feasible market for MERS-CoV vaccines, and that's why we haven't seen, and this is again an answer to people who say why we have a vaccine for COVID but not for MERS, because some of the elements are not there when you take the bigger picture. This journey, as we have heard yesterday from AstraZeneca, usually would take 15 years, but this year for COVID it was shortened to less than 12 months because all of the element is there, the need is there, the push is there. The discovery usually is covered by academia, while the phase 1 and preclinical is usually done by research centers or spin-out companies coming from academia. While the remaining phases up to the marketing this is usually covered by big pharma. And if you look at the case in Saudi Arabia, you would, we identify 2 gaps based on the previous and current effort. The GLP for animal non-clinical studies is lacking, and the current GMP small-scale manufacturing is also lacking, especially for R&D. You would find GMP manufacturing in Saudi Arabia, but not for R&D purposes. And that's where the biotechnology gap is in my belief. So I will just explain now 2 of the initiatives or efforts in Saudi Arabia, one in K-Mark and one nationally. So in K-Mark, we embarked on establishing or linking our vaccine development efforts, benefiting from the ecosystem that exists in K-Mark. So we have vaccine and ID lab. Though this lab is benefiting from genetic lab, proteomic lab, immunological assays lab, as well as a core facility, all supporting whatever is done in the vaccine development lab. Again, the animal facility in K-Mark has more than, could house more than 8,000 animal in a single time. So that's supporting the vaccine development. Clinical trial services, we've heard from Dr. Geraisi today that K-Mark has already set up and have a record in conducting clinical trial from phase 1 to up to phase 4. And the clinical trial services in K-Mark, they have they cover regulatory affairs, they cover the quality control as well. Research quality, where we cover clinical trial quality assurance, and then we have clinical trial phase 1 unit that was approved by Saudi FDA and used to conduct a phase 1 clinical trial unit, which has all the manpower, trained manpower, nurses, pharmacists, and clinicians, and so on. The Department of Strategy and Business Development supporting that as well from their angle. And recently, KMARC has launched its Biotechnology Park to support picking up any of these molecules or compound or vaccine and pushing them towards prototyping and spin-out like structure. In the vaccine unit or in the vaccine ecosystem in KMARC, we've been collaborating with multiple partners, mainly Oxford University, but also the International Vaccine Institute in Korea, the WHO. CEPI has also supported us in the past and still on board, as well as other universities locally and internationally. One of the projects that we have done is the co-development of ChAdOx-MERS with the group in Oxford, where this vaccine has been tested in mice, in multiple models in mice, and in non-human primates, and was done. We took it further and tested it in camels and published the efficacy, safety, immunogenicity of ChAdOx-MERS in camels. We have recently submitted and got accepted into the Lancet Microbe for our phase 1 clinical trial, testing that vaccine. And this is, to our knowledge, this is the first phase 1 clinical trial being done in Saudi Arabia, and there has been so much of effort and infrastructure put into place by KMARC and National Guard Health Affairs to enable this and to drive more to come of phase 1 clinical trials. So currently we have some vaccine academic research in the pipeline, mainly for MERS or COVID, based on adenoviral or pox viral or Maraba vector. We have also a project on mRNA-based COVID vaccine. And we have other projects for camelpox vaccine and for Rift Valley fever vaccine. And these are academically oriented, but they would benefit from biotechnology ecosystem that will enable them to move ahead. For COVID vaccine in KMARC, we have embarked on three different projects in collaboration with King Saud or King Abdulaziz universities. And this is our ongoing in the preclinical or proof of concept animal testing. At the global level, we have collaborated, we have put a lot of effort during the pandemic with the Oxford University, with the Sinovac, with the Gamaleya in Russia, Gamaleya Institute, in order to support their phase 2 or 3 vaccine development. However, for various reasons, this did not materialize to work. For example, the Oxford vaccine, at that time, the number of cases in Saudi Arabia during the lockdown was not enough to take the ChAdOx COVID vaccine into phase 3 clinical trial in Saudi Arabia. The Sinovac has some concern raised by the Saudi FDA, and that's why it was not. But however, this unit and the KMARC overall has embarked on vaccine development efforts, not necessarily taking from bench to bed, but covering different steps, different stages of vaccine development. Now, at the national level, and this is my last topic, I would say, at the national level, we have heard of the Saudi Vaccine Biopharmaceutical Center recently in Saudi Arabia. This is a national project led by King Abdulaziz City for Science and Technology through their RBDC, with the company, with the Saudi Vax company involved as operator and hosted at KAUST. We've heard yesterday that this project is supported by the Industrial Development Center and the NIDLIP as well. So we hope that this is one of the things nationally that would close some of the gaps and enable our academic product to move into development, clinical development. Here in the middle, the SVBC, or the Saudi Vaccine Biopharmaceutical Center, is due to be completed next year, so less than a year from today. And in the same time, there is, the group with, again, we have heard yesterday from the Industrial Development Center that they are working towards establishing a CDMO in the biopark in KAUST. So these were some of the national efforts that we are all hopeful to see them a reality. The center is already as I said, is going to be completed next year. Some of the capital equipment, the core lab equipment already brought into the site. So this is the last slide. If I want to summarize what is missing in this pipeline in our current or local national landscape, then it is GLP animal infrastructure and ecosystem, GMP development for a small scale for phase 1 or 2 clinical trials, reasonably quick regulatory approvals, as well as startup ecosystem. We know the startup is not yet mature system in the kingdom. And if we talk about biotechnology in the summit, then biotechnology is a bridge that could close this gap here, building on the existing efforts that I just mentioned, some biotechnology parks or clusters, startup ecosystem, regulatory and industry support, as well as investment that would close the biotechnology gap in Saudi Arabia and would enable our academic institutes to move their product, whether vaccine or otherwise, into the clinical development. Thank you so much.
So thanks a lot, Dr. El-Arbi. I think it was very nice to also hear about the challenges, and we will come back during the panel about some of the challenges that you have mentioned and how can we maybe learn from it to advance in the future. So I'd like now to give the floor to Dr. John Henrich, who will give us some insight about vaccinations in flu, challenges and opportunity in the future. So the floor is yours, John.
Well, thank you, Jamila. Thank you for the kind invitation. I'm glad to be here today, if not in person. I'm going to talk today actually about RSV prevention for all infants and children. So sorry for the change in the agenda, but hopefully the topic will be of interest to everyone anyway. I am here representing Sanofi Pasteur. Let's hope this works. There we go. So first, a few words about respiratory syncytial virus, or RSV, which is a member of the paramyxovirus family represented by two strain types referred to as A and B. The virus itself has two important proteins on its surface, the G protein and another protein which I'll talk about in some detail today, which is referred to as the fusion, or F protein, which is the target of both the vaccine and monoclonal antibody approaches from most companies. The virus itself is responsible for causing respiratory infections that lead to this characteristic formation of a syncytium, or a fusion of cells together, which lends the virus its name. Now, RSV is a common respiratory virus, as you heard from Jamila a few moments ago, really at both ends of the age spectrum. So in infants and young children, it is responsible for lower respiratory tract infections, most commonly characterized as bronchiolitis and pneumonia. And in fact, in this population, it is the leading cause of both bronchiolitis and hospitalization in children. In the elderly, in addition, it causes disease in for example, exacerbation of asthma and COPD, and is also responsible for significant morbidity and mortality in that population. But let's focus on the infant space for a little while, and I'll talk about that exclusively today. Here are some numbers on RSV in infants and young children. Overall, globally, RSV is responsible for over 2 million hospitalizations per year. In the US, that's about 33,000 hospitalizations. But really that number, oops, I don't know what happened there. That number of hospitalizations is dwarfed by visits to emergency departments and to pediatricians. And RSV is really the largest cause in typical years of disease that really puts a burden on hospital, on intensive care units, pediatric intensive care units, on physicians' offices in the winter months. Now, what we typically see for RSV is shown in the graph at right. And the gray lines are typical RSV seasons that occur in the temperate regions of the world between about November and about March. This was, of course, all turned on its head this year because of COVID. And what you can see is the incidence of RSV disease in green in the 2020 to 2021 winter season. And we really didn't have any RSV disease in many countries. This is largely due to the fact, of course, that people were staying inside, not sending their kids to schools or daycares, and taking precautionary measures to prevent COVID, such as mask wearing. What we did see, and quite to our surprise, was a peak of RSV infections that occurred in these regions in the summer months, and is really happening right now. So RSV, which had been a very predictable disease pattern, has been somewhat unpredictable this year. I will mention that this temperate pattern of disease in the northern hemisphere is somewhat different in tropical regions and in other parts of the world, where we tend to see a little bit more consistent RSV patterns throughout a longer period of time. And in fact, in places like Japan, we tend to see a prolonged RSV season that can last about 8 months in duration. Now, RSV will infect nearly every child by the time they reach 2 years of age. And of those children that are infected with RSV, nearly 1 in 5 will need to visit their doctor for diagnosis and treatment in the first 6 months of their life. Of those children that are infected, both 1 in 33 will become hospitalized in the first 2 years of their life caused by RSV. So clearly a huge burden, and one that is currently not met. Now, RSV, as I mentioned, is the leading cause of hospitalization in infants. This data is from the US, where we see about a third of all hospitalizations in kids under 1 year of age are caused by RSV bronchiolitis. However, if you look at that even more closely, about 40% of infections are undiagnosed with RSV, but are consistent with RSV infection. So a substantial burden of hospitalization is caused by RSV. Now, really important for people to understand, and one of the take-home messages from my talk today, is that the majority of hospitalizations that are caused by RSV occur in otherwise healthy infants that are born at term. This is a little bit conflicting with what people generally think of as RSV infections. We tend to believe that RSV is a severe problem in the premature and children with other comorbidities. And there is one prophylactic product that I'll talk about a little bit in depth that is restricted for that population. But that really means that about 80% of hospitalizations caused by RSV, we currently have no drugs to treat or prevent. So I mentioned before that RSV really has these two important surface proteins. The first of these is the G glycoprotein. I won't talk about that in any detail, but the second is really an interesting protein referred to as the fusion or F protein. And there was really a breakthrough in the understanding of RSV pathogenesis when the crystal structure of the fusion protein of F was solved. And it was well understood that the protein exists in two conformational states, both the pre-fusion and post-fusion forms of the protein. And that this protein undergoes a conformational change when the virus attaches to its receptor on the host epithelial cells. And this conformational change allows the virus to be brought into proximity to the epithelial cells and provides a mechanism by which the membrane of the virus and the host cell membranes can fuse and the virus can enter into the cell. And if you can block this fusion event, you can block viral entry and prevent disease. Since the understanding of the crystal structure of this molecule, it has become understood that the majority of neutralizing antibodies are targeting the pre-fusion form of F protein. And with that understanding, a lot of companies got into the race to develop both vaccines and prophylactic monoclonal antibodies for RSV prevention. Now, there has been a long history of attempts to develop vaccines for RSV. Really, this all started in the 1950s with this discovery of the virus, and subsequently a vaccine was brought into clinical development in the 60s, which was a formalin inactivated whole cell vaccine, whole viral vaccine. This immunization study led to an unforeseen consequence when those infants that were vaccinated became more susceptible to RSV, and a number of infants died following RSV infection. And that really put a hold on development of RSV vaccines for the better part of 30 years, I would say. In the 90s, the first prophylactic products for the prevention of RSV were developed. The first of these was a pooled polyclonal product referred to as Respigam. And then in 1998, the first monoclonal for the prevention of RSV infection, Palivizumab or Synagis, was licensed. And that's really been the only option for RSV until current times. Oops. Now, I mentioned Palivizumab. You know, this is a humanized monoclonal antibody. It binds to a highly conserved epitope actually present on both the pre-fusion and post-fusion forms of F. And it's been shown in two fairly large clinical studies to prevent hospitalization, either in premature infants or in infants with chronic lung disease or congenital heart disease. So we've had this product for 20 years or so. Why is there a need for better products? Well, there's a couple of limitations for Palivizumab. One is that it's dosed on a weight basis, so there's complications in delivery. And in fact, infants that are more than 7 kilos in size require 2 injections. But the biggest problem is that this is a typical monoclonal antibody, and because of its half-life, infants are required to receive an injection once a month during the RSV season. And therefore, it is restricted to use in only the very highest risk individuals. It's very expensive, and in the US, the indication is for infants less than 29 weeks gestational age, or infants with other comorbidities like chronic heart and lung disease. Whoops. Now, I'm going to tell you first about Nirsevimab. Nirsevimab is our collaboration between AstraZeneca, who leads the development and regulatory activities, and Sanofi Pasteur, who leads the commercial activities. And Nirsevimab is a fully human monoclonal antibody isolated from a patient with RSV infection. It actually targets a well-conserved epitope called the epitope 0 on pre-fusion form of F. But perhaps what's most interesting and most novel about this antibody is that it has been engineered to contain 3 amino acid substitutions referred to as the YTE domain. And the addition of these 3 amino acids allows for more potent binding to the neonatal FC receptor and recirculation of the antibody back into the bloodstream. That allows this antibody to provide rapid protection following injection and once per season dosing, because the extended half-life, as I'll show you, protects infants for a full 5-month RSV season. We've studied this now in 3 clinical studies with the endpoint of prevention of RSV-confirmed medically attended lower respiratory tract infection. And we hope to license this in the very near future for protection of all infants entering their first RSV season, and then children with chronic lung and heart disease entering both their first and second RSV seasons. Now, I'll show you the results of one study that we conducted here. This was a phase 2b study conducted in infants between 29 and 35 weeks gestational age in 23 countries in both the northern and southern hemispheres between 2016 and 2017, where a dosing occurred at the start of the RSV season. And in this figure, you'll see a little bit more information. The study was conducted in almost 1,500 infants that were randomized 2-to-1 to receive either a single intramuscular dose of Nirsevimab at 50 milligrams or placebo. We followed those infants for a full 150 days to assess medically attended lower respiratory tract infection, either inpatient or outpatient, caused by RT-PCR confirmed RSV. And we followed various secondary endpoints, including hospitalization, safety, PK, and health care resource utilization. Now, to be a case of RSV in the study, you had to have one at least RSV. You had to have the involvement of the lower respiratory tract, and you had to have some objective measure of clinical severity, such as increased respiratory rate or hypoxemia. And what I can show you in this figure is that in viewing the Kaplan-Meier curve at the top, you can see that there's very nice separation over that whole 150-day window between the infants receiving Nirsevimab and placebo. And that translates to 70.1% protection against medically attended LRTI caused by RSV and 78% protection against hospitalization. Maybe more interestingly, the antibody stayed above the target serum concentration, which was determined to be the effective concentration in preclinical models in 98% of those infants. And it actually stayed above that target concentration well beyond 150 days, suggesting that we may be able to demonstrate protection actually longer than a 5-month period. So with that data, we entered into 2 additional studies. The top study, MELODY, is a phase 3 study being conducted in 3,000 infants greater than 35 weeks gestational age. The bottom study is called MEDLEY, and this is a phase 2/3 study in which we compare Nirsevimab versus placebo in infants in there that are recommended based on local guidance to receive Synagis. And we're studying that in the first and second RSV season. And while I can't talk directly about those results today, I'm happy to say that we did meet our primary objectives in both studies. We showed efficacy, it demonstrated efficacy in the MELODY study, and we were able to show that we had similar safety and tolerability to palivizumab in the medley study. So with this, we are proceeding to file this product and move towards licensure. Filings begin in early 2022. Now, I do want to talk a little bit about our collaboration with the NIH to develop an infant vaccine, which we believe would be poised to protect the rest of infants prior to entering their second RSV season and be a follow-up to Nirsevimab. This vaccine is based upon intranasal delivery of a live attenuated vaccine of RSV that is called delta NS2. And the NS2 gene is an important gene produced by RSV that suppresses interferon responses as well as promotes virulence caused by RSV. So we have, with our collaborators, deleted this NS2 gene and provided two complementing mutations at positions 1313 and 1314, which further reduce the virulence of the virus and stabilizes the deletion. We have the advantage of being able to study this vaccine in the IMPACT network, and here Ruth Karen at Johns Hopkins University is our collaborator. And we can evaluate this vaccine directly in RSV seropositive children between 12 and 59 months of age, and then very rapidly bring these vaccine candidates down into seronegative and viral shedding. But we can also begin to see some hints of efficacy in this population. With this figure, this is a meta-analysis of multiple studies that have been conducted over time. And you can see that here we're focused on two candidate vaccines on the left. One referred to as Delta M22. The second is the Delta NS2 candidate that I spoke about. If you look, all of those produce very good neutralizing antibody titers when delivered intranasally. And they have various levels of shedding by PCR on the bottom left. We tend to look for viral candidate vaccines that have shedding below about 10 to the 5 PFU per mil. So you can see the NS2 candidate was selected based upon its favorable characteristics in terms of immunogenicity and viral shedding. Now, if you look on the right-hand side, this is some very preliminary efficacy data. If you look across all of these vaccine candidates, there is a strong reduction in both medically attended respiratory infections and medically attended lower respiratory tract infections, shown in gold. And if you only look at the candidates that produce neutralizing antibody, that protection gets even better. So admittedly, very low numbers, but some hint of efficacy that makes us confident that we can move forward with these vaccine candidates. So I'll wrap up here. I just want to remind people that RSV is the most frequent cause of respiratory disease and hospitalization in all infants. And while multiple approaches have been tried to target RSV, for the last 20 years, we've had only a monoclonal antibody, palivizumab, and its use has been very highly restricted. So 98% of the population goes unprotected. I talked about Nirsevimab. It's a fully human monoclonal antibody. It has increased potency compared to palivizumab, and the half-life extension technology allows us to provide a solution that should be able to protect infants for an entire RSV season. And finally, the live attenuated vaccines, we hope, will bridge those infants from protection with Nirsevimab to lifelong or at least early childhood protection based upon active immunization. And we have some preliminary results to suggest that our candidate is well tolerated and has at least preliminary efficacy. With that, I would like to thank you for your attention and thank all those patients and their families involved in the clinical studies, as well as my team at Sanofi Pasteur, our collaborators at AstraZeneca, and the NIH and Johns Hopkins. Thank you again.
Thanks a lot, John. I think this is a very nice example about two things. First, about the science that is required for developing vaccines. The second one is the time it takes. The vaccine for RSV had to pause for more than 30 years while understanding the biology of the virus, but also the biology of the right immune response to be induced before coming to solutions. And the last learning is that there is not one technology that fits one vaccine development. So having really a pool of technology to tackle those viruses is important. So now let's move to the next speaker, and our last speaker of the day will be Sven Free from the Danaher Corporation and Vaccines. So the floor is yours, Sven.
Yes, good evening, good day, everyone. Can you hear me loud and clear, I hope?
Yes, we do.
Very good. So my name is Sven Free. I'm General Manager for the Enterprise Solutions business of Cytiva. Enterprise Solutions being our end-to-end biomanufacturing solutions and enabling services that we bring to the industry in support of commercializing manufacturing processes and bringing products through clinical trials at all different scales. As many of you may not know Cytiva in details, I would like to share a few slides on our company, whereby our vision is to bring access to life-changing therapies to the world and by this transform human health through means of our by advancing basically those and accelerating those therapeutics through means of our enabling services, technologies and tools that we are offering to customers of all different sizes, academia and collaborators. Now, Cytiva is around for a long period of time, and some of you may know us as Emerson Pharmacia, GE Healthcare Life Sciences, and since the acquisition by Danaher last year in April, we closed that acquisition and a firm under the name Cytiva. We have been around the block for more than 6 decades, and Cytiva in particular is known for being well present in the downstream processing. Protein A is a very household name of ours, our Mapselect, Sure, and other type of family members here. But also even Nobel laureates have been working with Cytiva equipment in their laboratories as research tools, fostering their scientific results further forward. And if you look at key numbers here, more than 1 billion doses of insulin per year are manufactured with support of Cytiva materials, equipment and services, as are 75% of top-selling biologics, and 4 out of 5 approved, FDA approved cell therapies such as Kymriah or Yescarta, for example, are also utilizing our technologies and tools. Now, Cytiva, as said, being part of Danaher now. Danaher itself is a globally operating conglomerate focused on life sciences to a significant part. Danaher's market capitalization is about $230 billion as of today. And Cytiva, together with the companies that are depicted here on the slide, such as Paul, such as Beckman Coulter, Cyax, Leica, etc., we are forming part of the life sciences business within Danaher. Last year, this group of companies contributed about $10.6 billion in revenue to Danaher through an equal split across all major regions, US, Canada, Europe, as well as Asia, with more than 50% of the contributions coming out of the space of biopharmaceuticals. Now, looking at Cytiva in particular, we are at this point 8,000 associates around the world. We are operating out of many different sites, obviously manufacturing-wise, innovation-wise. 4 key hubs are to name, which are in the US, in the Boston area, which are in Sweden, which are in Bangalore, India, as well as in Shanghai, China, and we have significant activities ongoing with innovation and manufacturing, as mentioned. Now I need to shift the slide here. One key thing since arriving in Danaher, what has been very refreshing is the ability to invest into our future, to invest into the supplies for our customers, and in cutting-edge new technology developments that we can enable to bring to the market. Now, we've just recently announced about a $1.5 billion investment in the beginning of the third quarter, which is equally shared between Cytiva and Paul, if you wish, the Biotechnology group is in Danaher. And hereby, we are investing very significantly into new resin manufacture, expansion of our cell culture media, expansion of our single-use activities, but also general network improvements and enhancements in order to allow better products, faster turnaround times to our customers, and ensure key supplies for the future. Part of this mission is also hiring additional associates. So we are having about 2,000 associates planned to join the emerging Cytiva Paul Biotechnology Group team over the next 2 years. So a very exciting time for us as we are growing with the market and even beyond. Now, next step, I would like to zoom a bit more into the workflows that are coming from idea to injection, as we call them. So from vial to vial, if you wish, that we represent in our enterprise solutions offering. If you look at the upper right part here, you see this small line of equipment there indicated, which is essentially from a wave seed reactor through the main bioreaction down through the purification to the bulk API. That is our so-called Flex Factory. There's different types of platforms existing here, but the overall platform predominantly based on single-use technologies, partly introducing also third-party equipment. We have filtration technologies from our sister OpCo Paul, for example, and thanks to the acquisition of Venrex, which happened earlier this year, we do have the ability now to cover even the drug product part through aseptic robotic work cells that can help our customers filling personalized medicine, small-scale clinical trials supplies, as well as larger scale supplies for example, for vaccines, including mRNA. Now, the journey of our Flex Factories has been starting obviously in the antibody space, where the strong foothold of Cytiva resides. We have been fostering our technologies into the new zoo of molecules, as we call them at Cytiva, which is of course viral vectors, viral vaccines, adenovirus, lentivirus, adeno-associated viruses, etc. But also, of course, pDNA as a therapeutic agent, but also as a key element in the process of mRNA manufacturing that we have lately added, of course, also to our portfolio. Now, if you take this manufacturing platform, the Flex Factory, and put a modular building around it, we call it QBio. And there's different forms of QBio that are pre-designed modular manufacturing facilities that are off-site manufactured, brought to the site and deployed in a very record time through parallelization of activities. These QBios have been also evolving over time into different fashions. We have a standard antibody biosafety level 1 QBio type of facility. We added capabilities for biosafety level 2, viral vector manufacturing, cell therapy, for example, is a part of the offering, as well as lately viral vector manufacturing capabilities in QBio box, which are essentially clean room modules that can be put into a building shell. So it's taking advantage of existing building shells that we push these clean rooms into. And we, of course, have looked into adding also latest workflows such as pDNA, such as mRNA, which I will allude to a little bit later. From a timing perspective, Flex Factories take typically 9 months to 12 months from the contract and design freeze to the handover of the fully qualified train, while the QBio box type of offerings are in the range of 12 to 18 months. Similarly, rapid deployment and a very quick answer to production needs that we can provide here to our customers, thanks to platformization, standardization, and pre-design. This type of concept has been adopted widely in the industry. As you can see here from our track record, and we heard earlier, the track record, experience, know-how is a very critical element when you look for partners. We can bring this to the table. We have, you see, a very significant deployment in Asia, rapid growing markets that have no time but need fast turnaround. We have obviously in Europe as well as in the US, significant footprint in this space, but I'm very proud talking about the Middle East, North African region, where with SaudiVax in Saudi Arabia, we are deploying one flex factory in Tuval, in the KAUST University Center there. We are having flex factories in Egypt, for example, also engaged in viral vaccines manufacture for the Sputnik vaccine. Turkey is a place, and so there's multiple sites also in the Middle East that are leveraging Cytiva's flex factory platform technology, and we are heavily engaged, obviously, throughout the world in manufacturing processes for viral vector vaccine, viral vaccines, as well as other types of vaccines. Now, for today, I would further like to dive into the mRNA plasmid manufacturing solutions, as this obviously is a very actual topic that we see through the COVID environment. If we look into why are there mRNA-based vaccines and therapeutics, obviously, as the other speakers previously alluded to, there has been long work done in the mRNA space and the RNA space before. This is driven by increasing demand for novel therapeutics and vaccines, but the real breakthrough came obviously through COVID, where the rapid deployment of those vaccines and the broadly applied clinical trials led to a fast pathway into the market. Regulatory barriers could be overcome. The regulators were basically very positive on the results and safety that have been presented in these large trials, and Pfizer, BioNTech, Moderna, they have done a fantastic job in bringing these technologies to the market in this record time. Now that, of course, attracts the industry, and while pharmaceutical companies have been previously already invested in looking into this technology, we see lately a significant move from government organizations that obviously are looking for self-sufficiency of their country, that are looking into adding local capabilities, local short response times for any future infectious outbreaks that this technology, through its exhibits, lends itself to. So that's an area we see. Obviously, as mentioned earlier, also personalized medicine, digital biology plays a critical role here, and that's definitely a future for the mRNA place. Now, advantages are that high safety could be shown, reduced immune reactions, for example, through innovative capping technology. We see it's a highly conserved workflow, which allows a sort of a platform approach. It's widely used in cell-free systems, and there's obviously developments in the liquid nanoparticle formulation space that help to bring these mRNAs to the final target, to the cell where they're supposed to do their work. Now, if we look into processes, the blue dots are representing a typical antibody process, and the green dots are representing an mRNA process. Now, if you look at an antibody process out of a 2,000-liter bioreactor at the indicated titer, you see here up to 50,000 doses per batch for 100 milligram per dose assumption. If you look at a 50 times smaller reaction space for mRNA, you are able to produce about 50 times that amount of doses out of a 50-liter reaction space, which is quite dramatic, obviously, from the difference in numbers. Now, interestingly, through the exhibits of the nucleotides, you obviously see certain aspects in the downstream processing where some of these steps are growing in size, are growing in volume. So it's a bit of a counter current, if you wish, compared to the antibody space, where the process goes from big to small. In mRNA, you have certain steps that are quite significantly diluted. The lipid nanoparticle formation, the encapsulation, even requires solvents, which means specific specific requirements for equipment and facilities. So very critical to consider in the design and in the approach to these processes. Now, if we look at the mRNA manufacturing, obviously plasmid, especially when we talk about large-scale, pandemic-scale applications, is very critical as a precursor. Plasmid manufacturing is done through E. coli-based processes where the plasmid, the pDNA, is formed during the bioreaction. There's a lysis step, and through a series of clarification of tangential flow filtrations and chromatography steps, you yield the bulk product that, if it was a therapeutic, could be further filled into the final drug product. Or in case of the plasmid, obviously, we are, in case of mRNA, we are interested in a highly purified product that has, as possible, no host cell proteins, no host cell DNA, or very limited portions of that, endotoxin-free as possible, so that we can, with a very pure raw material or intermediate, go into the mRNA manufacturing, which is depicted here in the lower part. Now, if we look at key elements of such pDNA process, it's essentially a 7-day process from start to finish, from upstream through harvest to the downstream purification. And out of a 50-liter batch, you can get around 4.5–5 grams of plasmid, which you will see later on is relevant when we talk about a number of batches and number of doses that you wish to achieve. Cytiva provides a flex factory type solution here for this approach, and we are obviously having also pre-engineered QBO box designs that can be rapidly pushed into customer building shells. Going into the mRNA manufacturing, there's now, you see on the bottom of the slide, 2 different steps. One is the mRNA manufacturing itself, and then the 3rd step here is the encapsulation. The mRNA manufacturing finds place through a series of enzymatic reactions, where first and foremost, you are linearizing your plasmid DNA, and then use it as a transcript, if you wish, in the in vitro transcription to generate the mRNA that then, through specific technologies, is kept that goes through a series of tangential flow filtrations, resin steps to be formulated and bulk filled. Now, the mRNA itself cannot be easily deployed to cells as such, hence the encapsulation, which brings the mRNA to a lipid nanoparticle, is required using specialized equipment, significant amounts of solvents, as we mentioned before, and then through a series of further steps, purification steps, comes to a formulated product. Again, you can see the ability that Cytiva has, thanks to its equipment and technology platform, to resemble such workflow, including the encapsulation system from Precision Nano, which is an acquisition our sister OPCO PAL recently made and part of the Biotechnology group of Cytiva. And you see also in the end here the aseptic robotic work cell for filling activities, which is the SA25 from our Venrex affiliate. Now, similarly, as for the pDNA, the slide shows, excuse me, the slide shows the flexibility of the setup. So if you were to start with your mRNA workflow and encapsulation at a smaller scale, what this slide is supposed to depict is to show that with the equipment that you acquire, you can cover certain different scales even, and through select addition of individual units or scaling out of individual units, such as the encapsulation system or the fill system, you have the ability to address larger scales and effectively manufacture with almost the same process equipment the target molecules here, which shows how the flexibility of this platform is given and across different scales. Right, and this was the slide I wanted to refer to earlier. If you look at the mRNA production and purification activities, it's about a 4-day exercise. Encapsulation is another 1.5–2 days, and then filling takes a certain amount of time. Out of a batch at 50-liter scale, you generate, subject to efficacy or efficiency of this process, about 40 to 100 grams per batch, yielding quite significant amount of product per year. And you will see on one or of the next slides, what does it mean in terms of doses, that obviously everyone is interested in. How many doses can I generate? Now, again, as for the pDNA, we do have this, the ability to provide manufacturing platforms, as do we have ability to provide pre-engineered solutions in a clean room format that can be rapidly deployed for our customers. And now coming to the dose, if we look at the example of a 50 microgram mRNA per dose, an mRNA yield of 70%, a pDNA process yield of 30%, you would need about 130 grams of pDNA to generate 5 kilograms of mRNA, respectively 100 million doses. And this is sort of a pandemic scenario, population scale mRNA manufacturing scenario that can be deployed based on the equipment that we've been showing before. And with the scales per batch and the number of batches you can see here on the slide, in order to generate those quantities of materials for your population. I think very attractive platform, rapidly deployable, and if you take advantage of these types of manufacturing setups, our biosafety level 1 QBO box pre-engineered solutions for pDNA up to 200 liter scale, for mRNA up to 50 liter, and we are working on larger scales as well right now. This is an area where we can help our customers in facility design, in bringing Flex Factory, including all relevant support functions to your site in a rapid deployment. Now, with my last slide, I wish to conclude here. What we at Cytiva have been working out is through the knowledge of processes, through the materials, enabling services, as well as technologies that are at our fingertips, we are able to provide to our clients proven, pre-designed solutions that entail the manufacturing train, the heart of the facility, that potentially entail even the construction of modules, of modular clean rooms that are conducted off-site, and through the parallel approach with the manufacturing equipment allowed to reduce timelines, hence the fast deployment. When you enter with Cytiva into a production, to such production facility project, we have a fixed and firm timeline, we have a fixed and firm price, and therefore it's this very significant CAPEX and OPEX control that you can leverage. And the fast deployment not only comes through the very experienced people, but also by standardizing our commissioning qualification activities, which allow us to bring customers very fast to CGMP readiness, train them, and provide to them an advanced starting point in response to such pandemics and other general applications for therapeutic or other vaccine purposes. With this, I would like to conclude. I would like to thank the organizers for having us, having me here today. I appreciate very much also on the back of our earlier signing ceremony that we had on the MOU with KMARC. We are looking forward to further engaging in the kingdom through SaudiVax and other projects as well. And we are here at your support. And thank you very much for having us today.
Thanks a lot, Sven. I think it's also highlight the fact that we are discussing in a previous talk about innovation in technology for vaccines, but we should also highlight the important innovations in the manufacturing field in the vaccinology. My memories remind me that for polio manufacturing in the past, it took 8 to 10 years to have an up and running manufacturing. And now we see that with the technology that is also happening in manufacturing, not only you can decrease the time to have an effective manufacturing plant, but also all the tools that now being able through digitalizations allow to have a higher performance in manufacturing, so which is quite important. So I'd like to thank all the speakers for these very nice presentations that I would say give a good view about the history of vaccines, the success, the challenges. And I would like now that we open the panel for discussions and invite all the speakers to be part of the channel. So I just want to start by saying that vaccinology has proven to be very successful. We've been through several periods where, on one hand, tools has been available to eradicate some diseases, through hesitations because those diseases has disappeared, to a pandemic where we do see that the vaccine is one of the ultimate weapon to fight against those pandemics. We should be ready for the next wave. We know that it is a consequence of the climate environment change, the overpopulations on the planet, the increase of proximity with animals. So we expect in the next decade that this pandemic endemic will be coming more often than we would like. So what we have been through the last 2 years has taught us what should be improved, and also how we can be prepared at the planet level, and not only in some countries like we have done in the past, because pandemics were not as often observed. So today we are around how can we, as collaborators or key actors in the field of vaccines, being prepared for the future. So I will call out some specific themes and invite you to the discussions. And the first one is today is the capabilities. And I want to ask Dr. Naïef, what are the challenges that you have to have the right scientists, right capabilities to be able to nurture all the science that is needed to ensure that we are prepared for the next wave of innovations in the field of vaccinology. So I just want you to maybe develop about what are the challenges that you are facing in Saudi Arabia and eventually open the debate.
Yes, thank you so much, Jamila. Well, if we think about Saudi Arabia, then I think maybe it's different challenges than in general or globally. I don't think we have an issue with having talents, especially in the past 10 years. We have so many of scholars returning from across the globe with PhDs in biological sciences and vaccine or otherwise. And that's not going to be an issue. However, funding, management of funding, as well as some of the logistic issue for how to conduct the research itself. Also, there is another thing that I think is very important to mention, that academic institutions, they're usually free to do whatever research to a certain level. And there's no clear focus or consortium between different universities focusing on preparing for a next pandemic vaccine, for example, from now. So wherever you have scattered research across different universities, and that's in my mind, I think it's one of the issue. If we need, as a country, if we need to be prepared for the next pandemic, then we have to aggregate these talent or these academic institutions, establish a national program that they can focus on instead of just self-driven. And then to support that with a good amount of funding, but also better management of funding and providing all logistic support for research materials. Globally, I think it's different because globally we have a renowned institution, and we have seen in COVID, when COVID hit on, we did not see vaccine coming from every single Western country. For example, we haven't seen, especially for Europe, for example, Western Europe, not every single country produced a vaccine because there is this big pharma in the picture who are looking for a university or startup to pick up whatever vaccine, and everybody would pause or hold, leaving the way because these are the people who are going to do it fast. And we're talking here GSK, Sanofi, AstraZeneca, Pfizer, and so on. So I think in the global level, maybe more of prioritizing what technology to take. For example, in the WHO, they said, even before COVID, they said X disease has to be dealt with whenever it hits. And CEPI came to existence and made difference. CEPI made difference in terms of our preparation for vaccine development. And I think it just need more of these support proceeding along. That's it.
Yeah. Yeah, I think the pandemic has made people about the fact that there is no barrier now. This virus go across the world, so it's a problem of the world and not some countries. And the debate has been raised for many years when Ebola, flu or malaria, where there was a critical need for pandemic and support from the governments. And I would say the effort were done in some area, but not sufficient. And today we are really going through a wake-up call, which is how, as a nation, as a world, we need to be prepared in the future. So there is a lot of discussion going on in CP, WHO, but also in all the region of the world. So how can we get more prepared? And for something that for me, we have not yet tackled is equity about the solution across the world. And this is where industry and private company has to work to make sure that we are more ready in the future. Anyone that want to comment on this aspect?
So yes, Jamila. So one additional thing that I would like to add to what Naif was mentioning is actually the research ecosystem in Saudi Arabia have actually evolved over the past couple of years, and definitely over the past few years, we've seen an improvement in the ecosystem of research, from regulation, from research centers, advance in the technology, and definitely, as Naif was saying, actually the return of the scholar from abroad and the scientists that we have actually in Saudi Arabia have actually enriched this ecosystem. The need is actually a little bit different now. The need is actually the pharma industry need to come into this community of scientists and start working with them in actually investing in clinical trial and actually bringing clinical trial to the ecosystem here in Saudi Arabia. So it's not a lack of capability per se. There is actually capability that need to be built. That's absolutely correct. From a human factor, infrastructure, all those things, there is actually areas that need to be felt. But it's actually definitely maturing, and I've seen the maturation over the past few years. I joined the pharma industry not too long ago, so I was actually able to see how things are actually run on the other side as well. So I can tell for sure that in the past, at least a couple of years, I've been in practice for 10 years. So at least in the past few years, we've seen a huge improvement in the research ecosystem, and it continues to improve as we speak. So the need is actually now is actually a global international pharmaceutical company need to be present in Saudi Arabia more than before. That's additional thing.
Yeah, and I think that ecosystem is key. What has been done at KEMAR is quite impressive. And I do believe it's something that needs to continue to be leveraged and look for opportunity to further collaborate. So you have already a lot of technology, cutting edge, I would say even infrastructure. So definitely you are on the right track to have the right ecosystem to be able to work on the next innovations. I was asking a question about talent and pool of scientists, because we do face in Europe and in the US attritions because of the increased demand in the biotechnology field, biological field. So that's something that across some of the regions, it's becoming to be difficult to fight against talent. So I'm glad to hear that you don't have this challenge. So Sven, coming back to your last topic about manufacturing, we know that one key element in vaccine is all the regulations behind. Again, if you look about COVID, everything has been accelerated, but we know that that's not going to be the case for any normal vaccine development. But in the field of manufacturing, now that things has evolved, where there is more knowledge, do you believe that there is also a decreased timing to have facility approved by regulatory authorities?
I mean, surely there's a decreased timing in setting up these types of facilities. So when I joined GE Healthcare at the time, nowadays Cytiva, about 7 years ago, I had lots of discussions around stainless steel versus single-use technologies, leachables, extractables, versus standard types of approaches. And people have realized that by producing facilities and equipment in parallel, leveraging single-use technologies, reducing complexity of the setup, in particular around the bioreaction and the upstream part of the process, has led to much faster deployment of these facilities. So that's definitely a step where we, through lowering the capital entrance barrier, making available technologies to a broader amount of customers, has been increasing the ability to manufacture critical life-saving drugs for clinical phases initially, but then also fostering them faster to the market. With this acceleration, I would say, of course, regulators, they have been adopting continuously the standards at highest standard, but they have been also mindful about embracing these new technologies on clear evidence and on clear data, which the suppliers such as Santiva have been delivering over the years. So there's a high level of confidence in the meantime, good regulatory documentation, and we are working closely with our clients to support their filing processes and providing the relevant data that are needed to bring these approvals fast forward. Yeah.
Yeah, and I would expect, like for the mRNA, which is what I call the digital vaccines, because you are working with a platform, the regulatory path for any new product developed on that platform probably will be easier than some other platforms.
Absolutely. I mean, platformization plays a critical role in all of these. And if you look at antibody processes, they are highly platformized. We've seen, obviously, in some of the new zoo of molecules, as we say, some more variants. But on the other hand side, you have a typical backbone of equipment that may vary a bit in size. But after all, it's a critical backbone and platform that you normally can fall back to.
Yeah. So John, a question for you. So you nicely introduce a new way to approach prevention, which is monoclonal antibodies. So there is only few monoclonal antibodies available today for treat or prevent, I would say, disease. What are the challenges and how do you see monoclonal antibodies playing a role in the future of preventions?
Yeah, it's a great question, Jamila. You know, obviously, it's a fundamental change in the way we think, right? And we think that for RSV, this is really appropriate because in those very, very young infants, there's simply not time to develop an immune response before RSV season in many cases. So it is applicable there. I think, you know, the concept of using a monoclonal antibody as a vaccine for prevention for a whole class, a whole cohort of infants is something that the regulators are and the recommending bodies are really having to come to grips with. And it is a different way of thinking, and it challenges our whole process. So I do think that there is room, and I think that there are instances like we've seen not only with RSV, but with COVID now, where an antibody approach is very appropriate because it can provide rapid protection in an instance where you either can't develop an immune response or you don't have time to. So I think you'll see more monoclonal antibodies developed for infectious diseases and other targets in the future.
Thank you. So we are running out of time and I have still several questions, but I just want maybe to focus back on the COVID-19 and the research. So I have a question for you, Nayef and Hani. You mentioned about MERS. We know COVID-19 is not yet over. We know that coronavirus is likely to come back. Are you looking to work on the next, I would say, COVID pandemic? Is there any extensive research done in your center? And if yes, how would you, I would say, plan to further get prepared through surveillance to address any new coronavirus-like pandemic?
Yes, thank you so much, Jamila. We have continued to work on MERS-CoV and a few other universities in the country. And I think we have been producing a good amount of information that's needed. And some of these outcome from the research had helped to prepare for COVID, or at least have to deal with COVID. one of the things that I usually mention to my colleague physicians, I said, like, when COVID hit, at least our healthcare system, they know how to deal, at least psychologically, with a coronavirus patient in the ICU, regardless if it's COVID or MERS. But in the scientific level, we continue to work on the camel sector or a camel side. In 2019, we have produced a report that says 38% of camels brought for food production are positive for MERS-CoV. This is an alarming finding, which has a public health impact, which means the MERS-CoV could hit and cause a hospital outbreak at any time. Other colleagues from University and King Abdulaziz University, Jeddah, they actually produced a paper that's saying the MERS isolates, so a strain circulating in Saudi Arabia, are genetically different from the MERS coming from imported camels from Africa. And their conclusion was saying, for some reason, the MERS-CoV strain in the imported camels do not establish themselves in the camel population for one reason or another. And there is still higher prevalence of MERS-CoV among camels in Saudi Arabia, more than the imported camels from Eastern Africa. All of these information, I mean, also that leads us to thinking of recombination. If you have MERS-CoV or different strain of MERS-CoV in camels circulating all these years, then we have to be cautious that at any time point they might have some new emerging coronavirus. So yes, the short answer, we are continuing in a number of research centers in the country, yeah.
Yeah, and I think there is a nice paper coming up about cross protections, so SARS-CoV-1, SARS-CoV-2, and MERS. So I think there is a lot to do there to understand the biology behind the cross protections, to really work like in the flu. Maybe the holy grail would be to have a universal coronavirus vaccines. And as much as we are learning about COVID-19, I think we need to go back on the learning from MERS, SARS-CoV-1, and eventually the other one, it will come. So we are out of time, so unfortunately we will have to stop our discussions. I really would like to warmly thank the panel for the discussions, and I was really also very pleased to be part of this summit, which has been of a high quality, and also a high opening learning on things that we could probably do furthermore in the future. So I'd like to finish by saying a warm thank you to the organizer, to the speaker and the panel for the discussions, and looking forward for having more opportunity to work together. So thanks a lot.
Thank you.
Thank you very much. It was our pleasure. Thank you. Great.